of the best way to understand how anything works is to see first of all how it looks like, right? So get a visible picture of that particular molecule, how it looks like. So in scientific terms, we are trying to understand the atomic details of these receptors. So you determine structures of these receptors at high resolution, and for that you need to generate large quantities of these receptors. So we recombinantly express some of these receptors, the ones that we are targeting, purify them, and solve their structures either by X-ray crystallography or single particle cryo-electron microscopy, which is kind of more preferred technique these days.
Hey, hi, welcome to Biotech Talks. Today with us we have Dr. Janesh Kumar, a senior principal scientist from NCCS Pune. He will be explaining to us about his research at NCCS Pune on ionotropic glutamate receptors. Now, these glutamate receptors play a very crucial role in our central nervous system, and dysfunction of these glutamate receptors leads to various neurological disorders such as autism and epilepsy. So not only why, but how Dr. Janesh Kumar is performing experiments in his lab to understand glutamate as a neurotransmitter will be explained by him in this episode of Biotech Talks, and I hope you enjoy it. Hello sir, how are you?
Good, thank you.
Yeah, so you work on ion channels and transport proteins. Yeah. So can you explain what are those?
Yeah, so to explain that, I would have to take a step back and first tell you about cellular membranes. So we all know that cells are surrounded by this lipid bilayer, and even the internal cellular organs are also protected by this lipid bilayer. Now what this lipid bilayer does is that it not only defines the boundary of the cell but also protects the internal environment of the cell from the outside. And because this lipid bilayer is semi-permeable, it does not allow free transport of solutes or ions from outside to inside or vice versa. And so to overcome this problem, cells have evolved these receptors or ion channels or transporters that basically help the cell to intake or influx or efflux the solutes or ions or different kinds of molecules as the need may be. So ion channels are specifically membrane proteins, which are again embedded in these plasma membranes, and they allow, as the name says, they allow the influx of ions or even the efflux, and similarly transporters. So transporters can not only allow ionic exchange but also the exchange of various kinds of solutes. Right? Okay. So we per se are interested in glutamate receptor ion channels, and as you know, glutamate receptor ion channels are the major mediators of excitatory neurotransmission in our brain. So that means that they carry out approximately 60% of all the excitatory neurotransmission that happens in the central nervous system. And by excitatory neurotransmission, we mean that when action potential travels from one neuron to another neuron, at these specialized synapses, the action potential, which is an electrical signal, has to first convert into a chemical signal, right? Because the synapses are disjointed. There is no continuous connection between the neurons. And so the conversion of this electrical action potential into chemical signals is via release of neurotransmitters at these synapses. And the receptors that we work on rely on the neurotransmitters known as glutamate, right? So once the glutamate is released from one side of the synapse, that basically we call as the presynaptic terminal, and it goes into the synaptic cleft. Now on the postsynaptic side, you have the termini, other postsynaptic termini of another neuron, where these receptors are present, which we call as glutamate receptors, because they bind to glutamate. And in response to binding to glutamate, now these receptors can open and once they open, they allow influx of cations, because these are cation selective receptors. So this is what glutamate receptor ion channels do. They are ligand gated cation selective receptors.
So working on this, how is it going to help in healthcare applications?
Yeah, so as I said, the glutamate receptor ion channels are doing this fundamental job of like transferring information from one neuron to another neuron. So you can imagine that any dysfunction of these receptors, either in the activity of the receptor themselves, or their regulation and modulation by the other interacting proteins or partners, has been implicated in a variety of neuronal disorders, right? Starting from Parkinson's, Alzheimer's, or other neurodevelopmental or neurodegenerative disorders. So if you understand how these receptors work, what they look like, then you can develop and design strategies to kind of counter these ill effects of the receptor dysfunction. So that's what we want to do. We want to not only understand how these receptors work, but we also want to understand how we can regulate their activity. Can we design or develop molecules which can also alter its functional properties? And hopefully by doing those things, we may find suitable molecules that can help alleviate some of these problems, the neuronal and neurodegenerative disorder problems.
How do you actually do experiments in lab?
Yeah, so see, our lab works at the interface of neurosciences and structural biology. I am trained as a structural biologist, basically started by doing X-ray crystallography primarily during my PhD with Professor T.P. Singh at All India Institute of Medical Sciences, New Delhi. And following that, did a postdoc at NIH, where I got introduced to the field of glutamate receptors per se. And so in the lab we have to kind of, as I said, we look for studying not only the how these receptor functions. So one of the best way to understand how anything works is to see first of all how it looks like, right? So get a visible picture of that particular molecule, how it looks like. So in scientific terms, we are trying to understand the atomic details of these receptors. So you determine structures of these receptors at high resolution, and for that you need to generate large quantities of these receptors. So we recombinantly express some of these receptors, the ones that we are targeting, purify them, and solve their structures either by X-ray crystallography or single particle cryo-electron microscopy, which is kind of more preferred technique these days. And once you have the structures, the structural analysis itself can tell you a lot about how these molecules might be functioning. But then what we also try to do, we also try to trap these receptors in different states of activity, right? And we do that by saturating the receptors with different ligands, which are basically outcome of years of research in the field. So we know that this particular ligand can trap the receptor in closed state or can trap the receptor in desensitized state, things like that. So we trap the receptor in different states of activity, determine the structures, and then compare them, right? And this comparison can sometimes give you ideas about the conformational changes that might be taking place when the receptor cycles through different states of its activity. So that is one thing. So you know how the receptor might work, and then because you have these high resolution information, you know where the ligand binds, what are the various interfaces that are available in the receptor. You can also go ahead and design molecules that can basically disrupt this activity, disrupt the interfaces, or stabilize the receptor in one particular state of activity, things like that. So all this ultimately can affect receptor function in vivo. So that's what we try to do. So basically membrane biochemistry is one major area, then structural biology, and then functional analysis we do by electrophysiology. So because these receptors, as I said, are ion channels, and flow of ions is current, right? So you can basically record how these receptors conduct currents, right? What are their functional properties, basically. So that characterization also we do in the lab.
So how do you study its function? Like, do you incorporate in a cell?
Yeah. So see, as I said, the function of these receptors is to conduct ions, right? And in the neuronal setup, obviously that will be ideal thing to do. You want to look at how these receptor functions in neuronal milieu, right? In that environment. But then that environment is also very complex, right? There are several other ion channels and proteins and regulators which might manifest into the functions that will be difficult to analyze, right? So for that reason, what we do, we express these receptors in heterologous expression system, like in HEK cells or other cell types, and there then we do the functional recording by electrophysiology. And now because these other variants are not there, you are able to dissect out the function or the biological property or functional properties of these individual receptors without complexity. And then what you can do, as and when you know that these are regulators, you can also incorporate those regulators in your assay system one at a time and see how it is affecting their functions. And obviously structure based mutational analysis is also a thing that we do. So based on structures, when we identify which are the critical residues that seem to play important role, we validate them by functional analysis. We mutate them and look by electrophysiology whether actually it is affecting the function or not.
How do you actually insert those receptors into cells?
See, when you are recombinantly overexpressing them, so basically you are doing transfecting the cells with plasmids, right?
Okay.
And these plasmids express and then they automatically get inserted into the plasma membrane. Yeah.
Okay.
But yeah, if you're talking about there are ways one can even break it down further, make it even more simpler. So you can argue that even in HEK cell there might be so many complexities, right? HEK cell itself has several other endogenous channels and receptors, as I told you about the neuronal system. So even there are ways by which people can make just lipid bilayers. So these are called bilayer BLM recordings or bilayer recordings, where you can just have these bilayers and reconstitute your purified receptors in those bilayers and record. So that actually just tells you about that particular receptor, free of all the other complexities that might be there. So see, in science, reductionist approach has helped decipher several of these complex problems, right? We nowadays talk about looking at systems biology, looking at systems as a whole. Yes, that is important, but then to first understand complex problems, you need to break them down into simpler parts and look them one at a time, then combine them together and then get a holistic picture. Yeah.
So you insert receptors into cells, and by measuring electrical conductivity you see whether it is functioning or not.
Yes. So there are all these receptor or ion channels can be defined by a set of parameters, right? So these receptors are like how fast they open, what is the amplitude, how good are they in conducting, right? And then once they are open, how fast they close, right? And how fast they desensitize, another term where the receptor can close even in presence of ligand, right? So one can, on the basis of these several parameters, one can characterize these receptors. And it is really fascinating that all these receptor subtypes which are there, they are functionally very distinct. So with a set of 4 or 5 parameters, even if you don't know what receptor it is, at least in our field, we can tell that this might be one particular receptor subtype and this might be second receptor subtype. So all these 4 or 5 parameters can help you characterize the receptor functions.
Can you please elaborate more on how, by analyzing the electrical conductivity of a particular receptor, its function can be deducted?
when we are talking about these glutamate receptor ion channels per se, so we know that they allow the conductance of cations, right? And these are non-selective cation channels. So you must have heard about sodium channels or potassium channels, which are voltage gated. These are ligand gated. Those are voltage gated, and there you know that these ion channels are very specific to either sodium conductance or potassium conductance, or in other cases like chloride channels, they conduct chloride ions. Glutamate receptor ion channels, on the other hand, are non-selective cation channels. So their pore is not selective to any one particular kind of cation. It can allow passage of sodium, potassium, calcium, things like that. So because this is like critical for its biological function, this cation conductance is critical for its biological function, you can actually see, as I said, you can characterize the receptor functional properties. And for instance, there are several SNPs which are known for these receptors, and if you make those mutants in the receptor and now compare it with wild type, then you can see how these four functional properties that you are measuring are changing between the SNP variant and your wild type, and that can give you ideas into like how the receptor functions are getting altered, right? And then you can do a screening of ligands which might have a potential to alleviate those functional changes as well. So it's an ongoing process, iterative process, where you do a recording and you make mutations, and then you go back, do recordings again, and you compare it with together with your structural analysis, and then that gives you a complete picture of how these receptors work inside the in vivo system. And yes, you can always combine it with animal model studies or cell-based assays, again to validate many of your findings which are from in vitro system.
So what motivated you to take up research on protein?
Yes, that's a very interesting question. So see, during my graduation and masters, I used to read, you must have also read biochemistry, Leninger and Voigt, and there you see these structures, beautiful pictures of structures of proteins, right? And even that time, I used to think, wow, this is so fantastic, right? Because a picture speaks 1,000 words, right? And in this case, you have the whole three-dimensional structure, right? So you know each and every atom, where it is positioned, how it is structured. And that was very fascinating for me, because these are such small molecules, nano machines, you cannot see it, but there are techniques and ways in which you can go ahead and get a magnified view, or you can determine the structures of these proteins. So that was fascinating for me. Ion channels per se, I got interested in around 2003 while I was doing my PhD at AIIMS. So if you remember, at that time itself, Rod McKinnon got the Nobel Prize for his potassium channels, right? And I happened to read that Nobel lecture. Couldn't see the Nobel lecture at that time, but read it, and I was completely fascinated by this whole idea of how these ion channels can impart selectivity to potassium, right? And see, the difference between potassium and sodium is so minor if you can look at their radii, and still these ion channels are so specific, 1,000 times, 2,000 times specific to potassium versus sodium. So that fascinated me, and so after my PhD I started looking for positions to get trained into this area of ion channels, and happened to join NIH with my postdoc mentor, Dr. Mark Mayer, where I got introduced to glutamate receptor ion channels, and so the journey started from there.
It was really interesting to know.
And see, that's why it's really important as a student to kind of be aware of what are the latest scientific discoveries that are happening around, and read as much as possible, not only your area of research, but also go outside your area of research and look at different things, attend talks which are not only specific to your area but also other areas. And many times these non-related areas, when you listen to talks or discuss with people, you get ideas that you can implement in your research. Yeah, and many of these things can be like life defining, because that moment was life defining for me. That because of that reason I went to pursue ion channels, membrane proteins, and that's why my lab is here. I'm here.
Thank you very much, sir, for your time.
Yeah, welcome.